Thank you, everyone. My name is Shayan Langha, and I'm the America's TruSight Oncology 500 Marketing Manager. I'm going to be going through a ton of information here, so we'll try to give a good 5 to 10 minutes for Q&A towards the end. Otherwise, you'd be able to catch myself and the Illumina team at booth number 13 on the right-hand side there. So with that being said, we can get started. And by the way, could you hear me, or should I get this close to the mic? This is better? Okay. Okay, so this is our agenda for today. I'll highlight Illumina's mission. From there, we'll touch on the oncology testing portfolio, which includes a myriad of products that many of you are well familiar with. And from there, we'll transition into our suite of connected software products, the support we offer our customers. Okay, so our mission at Illumina is to improve human health by unlocking the power of the genome. Our sequencers enable researchers in academic, government, commercial settings to comprehensively understand the genome and how it translates to health and disease. Our technologies empower healthcare providers to leverage those genomic insights in clinical care, providing patients with diagnoses and groundbreaking therapies. Okay, at Illumina, we're developing solutions for all customer types and all testing opportunities within the oncology testing journey. On the left here, you'll see the myriad of panels we offer for solid and soft tumors. On the right, you'll see the different flavors for heme tumors. So starting with the left, we offer library prep kits for customers that are looking to start their NGS journey, with targeted panels and those that are looking to take advantage of larger comprehensive genomic panels. You can see the portfolio is integrated with our partners Pillar Biosciences, where we offer 4 distributed kits. We offer Pillar OncoReveal Multi-Cancer CNV plus RNA Fusion, which is an RUO product. Pillar OncoReveal BRCA 1 and 2 plus CNV. That is found under the small somatic and germline panel category. You'll also find Pillar OncoReveal Myeloid Panel and Pillar Essential MPN. I'll go through the panels a bit further in detail in subsequent slides here. A quick question for IT here. So like I said, I'll go through the pillar panels in subsequent slides, touching base on high-level information you should know about them. With that being said, I'll also provide some insight onto TruSight Hereditary Cancer Panel here at the booth if there's additional questions for that. But know that our portfolio also includes TruSight Hereditary Cancer RUO and Infinia methylation. There is a new guideline-directed test with CNS methylation, often Array, which is currently helping to reclassify many misclassified tumors, which leads to more accurate therapeutic guidance. And it is the same thing with hereditary cancer testing and NGS somatic testing as well. Now, for those customers that are wanting to run a comprehensive genomic panel, they're able to do so with our very successful TSO500 tissue and TSO500 ctDNA assays. But even with all these panels listed, we are continuing to build our portfolio, and the next that will be added will be the FDA-approved OncoReveal CDX solid tumor IVD, which has been validated and approved for use on Illumina's MySeq-Dx system. This panel covers 22 clinically relevant genes. It has a single-day workflow and a sample-to-report time of as little as 48 hours. It's not presented on this slide because we're not distributing it just yet, but feel free to reach out to myself or your respective pillar partners, and we can get you additional information on that. So moving on, you can see the full scalability and flexibility that Illumina offers for the oncology testing journey. You can see the plethora of different products and what platforms our solutions are compatible on. From low to mid to high throughput, Illumina offers that true flexibility and scalability for each customer's lab. Just keep in mind, we'll be discussing DRAGEN's secondary analysis, which is available on-prem and ICA, alongside tertiary analysis options towards the end of this presentation. We can see TSO 500 is compatible with NextSeq 550 and TSO 500 high throughput on the NovaSeq 6000. It's not presented on this particular slide, but TSO 500 high throughput plus TSO 500 ctDNA version 2, which we launched last year, is now available to run on the NovaSeq X series, where you're able to decrease the cost per sample while taking advantage of many other benefits on the NovaSeq X platform. In addition to this, Illumina has demonstrated feasibility of running TSO 500 high throughput plus or minus HRD libraries on NextSeq 1000, NextSeq 2000, and the NovaSeq 6000 systems. I will provide a nice peek here behind the curtain and say that we do anticipate a big announcement soon on TSO 500 high throughput, launching on NextSeq 1K and NextSeq 2K very soon. So please keep a lookout for that. Oh, can we go back one slide? Okay, so we all know this. The TruSight Oncology portfolio has been incredibly successful since the inception of its launch several years ago. But why is this? Why do our customers consistently come back to us, and why are labs constantly reaching out? Well, we offer a sample-to-results workflow which consists of an automation-friendly kit, out-of-the-box bioinformatic pipelines, and integrated sample-to-results with the turnaround time between 3 to 5 days. We have expertly curated content with a pan-cancer panel design, 523 genes that cover key clinical trials and guidelines. Interpretation report is delivered via ICI or Illumina Connected Insights, or Valsera's CGW, or even other commercially available platforms. TSO 500 is also analytically validated, where the performance data is strong. It has high analytical sensitivity and specificity, with automation partners that I'll be highlighting a bit further on. There is also an IVD roadmap that is currently in development and built upon the TSO 500 portfolio content. This is also pharma endorsed, which allows our customers to be confident with their choice of assays and allows for an easy out-of-the-box implementation. Okay, so there's a lot on this slide here. I'm not gonna go through everything here, but I do wanna highlight a few points. TSO 500 high throughput and TSO 500 ctDNA version 2 are both compatible on the NovaSeq X and NovaSeq X Plus. If you take a look at the sample throughput row here, you'll see the different flow cells. So under TSO 500 high throughput, you can see that on the 1.5B flow cell, labs are able to run up to 32 samples. On the 10B flow cell, up to 192 samples. And on the 25B flow cell, 480 samples. When you take a look at ctDNA version 2, which we launched with last year, which is the second iteration of our successful ctDNA version 1 product, you'll see 1.5B flow cell can run up to 4 samples, and the 10B flow cell runs up to 24 samples. A few other noteworthy items I want to highlight is the content is the same as tissue. The panel size and length is the same. They're all across 1.94 megabases. One item that makes ctDNA version 2 the differentiating factor amongst other market players is the high sensitivity this product offers. So you can see at just 20 nanograms of cell-free DNA, which is the recommended amount, you're able to attain 0.2% VAF for SNVs at 90% sensitivity. And we also have data that showcases superior performance between 5 to 20 nanograms of DNA. I also wanted to highlight TSO500 HRD, where we in-license Myriad Genetics' GIS algorithm. This allows for the interrogation of 25,000 SNPs across the chromosome to help identify the 3 genomic scars: LOH, or loss of heterozygosity, TAI, or telomeric allelic imbalance, and LSTs. With the addition of HRD on TSO500 RUO, labs are able to gain more efficiency and thus use less sample when compared to running a separate HRD assay. Other market players assess between 5,000 to 7,000 SNPs and don't come anywhere close to the resolution Illumina has with over 25,000. We can also see a very high 96% agreement rate for TSO500 HRD versus the gold standard, and a 0.98 concordance rate for TSO500 HRD with Myriad's GIS. TSO500 HRD is a true CGP solution that allows labs to integrate genes in the homologous recombination repair pathway, the 3 genomic scars based on the high resolution we have, BRCA1 and 2 large rearrangements, to attain the HRD status for samples. This is also a very nice illustration that highlights the no increase in turnaround time or DNA input, which stays the same. The TSO500 DNA library is split into 2 parallel enrichments in the same plate. So one for DNA panel and the other for HRD, and you can see that here, the orange. And this ultimately allows us to integrate HRD into our TSO500 panel. So this next slide is our latest DRAGEN pipeline update for TSO500, version 2.5. The first item you see on the left here is we're extending intronic variant calling to 10 base pairs. Before we had 2 base pairs, so this will allow to detect more biomarkers. For example, European customers have been asking to add a Belgium founder mutation in BRCA1. That was 3 base pairs into the intron. Now that's covered with this. Second is we've expanded the copy number variant testing. Before, we only had amplifications in 59 genes. In this release, we expanded to 514 genes and added deletions. These 2 additions were frequently asked by customers and will greatly expand the scope of biomarkers that we can now test. Third, sex prediction. You may know that sex of the test subject is often determined bioinformatically by labs in order to confirm that the samples are not swapped. Our customers now do not have to run an extra algorithm because it is available in the same to run. It's very convenient. So these 3 new features are available for TSO500 mid throughput and TSO500 high throughput. The next 4 features now, tumor fraction, ploidy, absolute copy number, and loss of heterozygosity or LOH, are available with the TSO500 HRD as beta features. What this means is Illumina has done limited or no testing of these features. However, we have a great degree of confidence in beta features because they are based off the validated Myriad GIS algorithm. When you take a look, when you take a look at the way these beta features are listed, in order for labs to access absolute copy number and loss of heterozygosity, in the algorithm, tumor fraction and tumor ploidy have to be calculated first. Another important note I want to highlight is we will provide gene-level loss of heterozygosity. Specifically, we will report which genes overlap with LOH events in the genome, which will help understand the impact on these genes. Okay, so TSO 500 is great, but what about the labs that are looking to automate and reduce their hands-on time? Illumina offers an end-to-end solution with our automation partners listed here. So you can see Beckman, Hamilton, and Revity. You will also see our suite of TSO 500 products on the left-hand side, and you'll also note whether each of these products have an Illumina qualified method or a vendor-developed method. The newest automation method I want to highlight here is TSO 500 ctDNA version 2 with Hamilton's NGS star MOA. So if there are questions on that, feel free to reach out to the Illumina team and we can help you get any clarifying insight into this new automation method. And this is very recent, as of 2 to 3 weeks ago. So that was TSO 500. But what about labs that are new to NGS or those that are looking for targeted panels and want to ease their way into a next-gen sequencing panel? This now brings us to our partnership with Pillar, where you can see kits that we currently distribute. I'll go through each of these later on in the presentation, but you can see they're all listed here on the left-hand side. There's 2 index kits, a combination of 32 and 96. And this is important as it provides flexibility for a lab depending on their throughput and the number of samples that come in. These assays can be run on Illumina's MiSeq or MiSeq DX system in RUO mode and NextSeq systems. And you'll see here some secondary and tertiary analysis options, which we'll highlight a bit later on. The first panel, Pillar OncoReveal Multi-Cancer CNV and RNA Fusion. You can see this panel covers variants in CNVs detected from both DNA and RNA, with everything you would need for lung cancer. Other market players have ALK DNA only, and you can see that we offer both ALK DNA and ALK RNA, which is a major differentiator. This panel, again, is run on Illumina's MiSeq with great plexity, which provides cost-effective options. There is a sample to answer in 48 hours, and that includes an 8 hours of assay runtime. This panel requires very low DNA input and can be used in the case that formalin-fixed paraffin-embedded tissue is compromised. So for example, if TSO 500 isn't sufficient and based on DNA input, you are able to run on one of these 4 distributed panels we offer and get results in under 48 hours. Panel number 2, Pillar OncoReveal Myeloid Panel. This panel performs well with internal tandem duplicates. You can see genes on the left-hand side here and note CEBPA, which is a very GC-rich region. Pillar OncoReveal has no problem calling this, and this is something other market players deeply struggle with. Panel number 3, Pillar OncoReveal Essential MPN Panel. They cover 3 genes, MPL exon 10, JAK2, exon 12 to 15, CALR exon 9. Usually, labs do this with PCR, and it becomes an iterative process because you're waiting on results on one test before going to the next gene. This increases turnaround time and prevents any delay on actionable insights providers are looking for. With the Essential MPN panel, you can get results with our single panel without iterative testing or waiting for results. Another item to note is this is a panel that no other market players offer, and the total turnaround time here can be under 12 hours because it's only 3 genes, and this is one of our highest selling Pillar panels we distribute. Okay, so this is Pillar panel number 4. BRCA1 and BRCA2 plus CNV panel. They're big regions to sequence and other panels aren't as comprehensive to capture this, but with this panel, we offer a full CDS with flanking intronic regions, 283 amplicons, and of course, it's great for prostate and breast cancer. Okay, so that's our library prep solutions. We'll transition into our suite of Illumina connected softwares now. No matter which library prep solution you use, we have the informatics to match on the analysis. We can provide sample tracking through Clarity LIMS. We offer a base-based sequencing hub to run and sequence samples. We offer DRAGEN and on-prem. We offer DRAGEN and on-prem and through Illumina Connected Analytics for secondary analysis. You're able to get insights from a myriad of applications, including oncology interpretation from ICI. You can see Partech for multiomics discovery, Imagine for genetic diseases, and much more. Our professional services team can also interface the complete software suite with your EMR, giving you a full end-to-end solution from sample accessioning to results and reporting. DRAGEN. So DRAGEN for secondary analysis is applied to oncology research applications. DRAGEN was designed as a generic software that can support the whole range of somatic use cases. DRAGEN provides support for different sample types from FFPE to liquid. This leads to many applications that are shown in the slide. TSO 500 and Pillar panels are fully supported as well as other CGP assays, as long as they are based on enrichment or amplicons. You can see MRD and monitoring for whole genome sequencing, tumor normal workflows for MRD testing, therapy release monitoring, response monitoring. There's solid tumor for whole genome sequencing or panel agnostic bioinformatics workflow. We have germline identification for hereditary cancer marker and mosaic variants. There's capability with DRAGEN for methylation variants, heme risk stratification, and liquid biopsies, and this is what makes DRAGEN top tier. DRAGEN generates the most accurate list of variants detected in both small and large oncology panels, but that's only half the story. What the presence of these variants in the tumor have to say about the sample is really what you're after. And we have you covered with our tertiary interpretation software, Illumina Connected Insights Research. It sorts the variants by genes of interest, given the tumor tissue of origin or cancer type, and looks up relevant agency guidelines, drug labels, and clinical trials for each variant. The output is pre-formatted in a report that you can review, edit, and augment to your exact needs. Connected Insights Research pulls from over 55 information sources, including JAX CKB and OncoKB. You can even plug in your own knowledge base to annotate with your curated information about the variants that are found in the panel. Okay, so by assembling a complete NGS workflow from Illumina, users are able to get significant advantages. Number one, a fully automated no-touch workflow from Illumina sequencers that excludes manual data transfers or other file manipulators. Number two, out of the box compatibility with the latest DRAGEN pipelines, or you can use third-party tools. Number three, the entire workflow can be implemented and supported by one team. And all steps come from one provider, so all data can be consolidated in one cloud, streamlining data storage and oversight. When set up, Connected Insights Research can automatically ingest samples and subject data coming from LIMS and EHR systems. Output from Insights can also be delivered back to LIMS and EHR. All these advantages will make it easier for the lab to streamline current tests, as well as adopt new assays and applications in the future. So what makes Illumina a trusted leader? We put our customers first, making them heroes, and provide the best service and support across the globe. We have over 400 service and support team members, which span from informatics and IT specialists to field service, application, product specialists, a team for customer success implementation, and much more. And this is the reason why Illumina is a trusted leader in DNA sequencing technology with that one mission to improve human health by unlocking the power of the genome. Thank you. So we have a few minutes here for Q&A, but also note if there's questions that we're not able to fit within the next few minutes, we're right across here just on the other side at booth number 13. So feel free to drop by, say hi, and converse with myself and the rest of the Illumina team. So just to make sure I understand the question.
The sample swap QC that you spoke about, where you can distinguish if the samples are swapped.
Yeah.
I assume you have a panel of polymorphic SNPs in there where you check for SNPs between your tumor and normal samples. I'm curious, how many SNPs do you have in there?
Okay, so that is a good question that I personally don't have insight for. That's DRAGEN version 2.5 we're talking about. Do any of you guys have insight into that by any chance? No? Okay. So what we can do is, after the talk, just contact me and then we'll provide you information on that and we'll get a deeper look with some of the product specialists for the DRAGEN pipeline side.
Yes, thank you very much for that rundown. So my question is to do with the pillar panels, especially the 3 gene for the myeloid. I like that idea. However, how cost effective do you think that's going to be for I'll say a medium-sized lab, where I saw your MySeq capability for plexity was like 192 samples. Was that what I was reading correctly or not?
Yeah, okay, let me do this. Would you guys mind going back to the slide so I can...
Because I wanted to know if it was cost effective for a medium-sized lab that's, that may be getting 50 samples per week or something to that effect.
Got it, okay. So you're right, the plexity is up to 192. And you mentioned a relatively low number of cases at this point, right, compared to the plexity that we offer for 192. So if that's the case, it probably won't be too cost effective just based on you trying to load up the flow cell. But I am not the pillar expert here. I'm the marketing manager for TSO 500 for the Americas. And this is a distributed panel where we have a separate team with pillar that is able to provide more information when it comes to cost effectiveness. So that's something that I'm happy to connect you with. That way we can help you figure out a nice solution for you. Mm-hmm. Any other questions?
Hi, Beth Patel, Mayo Clinic. I saw that you're integrated with JAK-CKB on the interpretation side. Recently, they were acquired by Mastermind Genomenon. I'm wondering if you have any insight to how that might affect your product moving forward.
That's a great question. We don't have insight into that right now, but let's connect and we can, see if there's some insight we can provide you with that. Yeah, thank you.
Hello, question about the DRAGEN. So currently it's only running on the FPGA machine. Do you have any plan to bring it to something other, more generic server?
Sorry, could you say that one more time?
Oh yeah, sure. The DRAGEN is right now it's only running on FPGA machine. So do you have any plan to use something equivalent or some of other solution for the lab without the FPGA?
I'm sorry, I'm having a really hard time understanding what you're
Yeah.
saying, but
The DRAGEN is running on FPGA machine, which is specialized server.
okay.
And so is there in the future, are you going to support other solution predates the DRAGEN? So something like you run on a generic server or other machine?
So when it comes to, like the bioinformatics pipeline and everything DRAGEN related, it's specifically for DRAGEN. So we would encourage our potential labs to look into that as a solution, but I'm not too sure. I really don't. It's tough for me to understand what the question is itself, but let's connect afterwards and I can help you out.
Sure.
Yeah, thank you.